Hello everyone, welcome to the problem solving session 7 for the course Cell Biology, Cellular Organization, Division and Processes. This course is offered by NPTEL, and like the past 6 problem solving sessions, the idea of this session is also to revise the topics that were discussed in the week 7 for this course, and then give you some questions from previous year assignment, exams, and some new questions so that they give you a overview of what the topics were and understanding of how they were connected to the previous topics. So in today's session also, you will have questions which are pertaining to topics that were discussed in the week 7. But we also have some questions that are from previous week just for building a connection. So let me begin. So these were the topics that were discussed in week 7. So the broad theme was regulation processes of cell cycle, mostly about the cohesines complex and its functions. We also did look into meiosis once again, mitosis also once again, and cell diversity and the properties of specialized cells with the case study of being from yeast. Okay, so like I said, before going into the topics which were part of the week 7, we will just do one of the topics that were in the previous sessions or previous weeks for building a bridge so that we get a continuity. And we will be doing or touching upon the topic apoptosis. So just for your revision, apoptosis, we have already discussed this in previous sessions in great detail. So it's just like that, like we just will revise once and then we'll go to the new topics. So just for your clarity, once again, apoptosis is a programmed cell death, which is occurring in a regulated and controlled manner. And because they are so important for development, tissue homeostasis, and also removal of unwanted cells and damage, that's why they are they're carried out in a regulated manner. Okay, so as you can see from the images here, so if you look into the microscope or if you observe the cell physiology when or cell anatomy when they are undergoing apoptosis, there are some key features. You will see cell shrinkage, you will see chromatin condensation, DNA fragmentation, membrane blebbing. So we discussed all of these in details in the previous sessions. We also saw formation of apoptotic bodies and then no inflammatory response. We had seeing the distinction between why there was no inflammatory response here, right? And on the right side here, it shows you the different pathways that are associated with apoptosis. So the intrinsic and the extrinsic. Intrinsic are basically, as you can see here, this part inside here, intrinsic. It's basically the mitochondrial pathway. These are activated by DNA damage, oxidative stress, or any problems with growth factor. And the protein family, which is responsible for the intrinsic pathway, is BCL2. And we saw how they are both the pathways basically converge at the step of like caspase 3 and 7. But the idea here is that the ultimate step is the cytochrome C release from mitochondria here. And then this activates the caspase 9, as you can see here. So it's, remember we discussed about apoptosome, mitochondria release, all of these, cytochrome C release. So all of that is part of your intrinsic, which is something which is inside the cell and driven by internal factors. You also have the extrinsic pathway, which is basically triggered by death ligands, or molecules that are specifically when they attach to the cell, they trigger the apoptotic pathway. So extrinsic is basically death receptor pathway, and the pathways are the molecules that bring in the information are called the death ligands, for example, FAS or TNF alpha, and that activates caspase 8. Right. So remember about the caspases, which caspases for which where for which pathway, and both of them ultimately converge at this step of 3 and 7 being the executioner caspase, if you remember clearly, right? Yeah. So these are topics that you should remember. You also should remember about the pro-apoptotic protein and the anti-apoptotic protein that we discussed. Pro-apoptotic being BAX, BAC, and BID, and anti-apoptotic mean the BCL families, BCL2 and BCL-XL. So remember which pathway is for like which proteins family are responsible, what kind of effects on the cell. So pro-apoptotic, anti-apoptotic, as the name suggests, right? And you should remember the receptor name for the extrinsic pathway, which actually bind to the death ligands, which are FAS for their FAS ligand, TNF receptor for the TNF alpha. So similarly, so there are some components that you should remember. But like I said, we have discussed this in the previous sessions. So if you have any doubt or if you want to do it once again, please go back to those sessions. Okay. Now, what I have done here is I put some questions here, which I have tried not to repeat from the previous sessions, just as a measure of revision. So I will go to the slides, read out the questions, and wait for 2 minutes, 3 minutes for you to attempt the answers. And once the like once 2 minutes are over, I will discuss the answers with you, right? So the first set of questions are, which process involves programmed cell death essential for development and homeostasis? What is the primary characteristic of cellular senescence? The accumulation of which type of cellular damage is a hallmark of aging? And which protein is a key regulator of apoptosis in the intrinsic pathway? These are the kind of questions that you will get for apoptosis in general. So just 2 minutes after that, I will give you the answers and also discuss them. So just a reminder, when you are trying to look into the answers here or when you are trying to answer the questions, try to think of the respective proteins. For example, in this question, it's very important which one do you answer. The answers look similar most often, but that's the key about MCQs. You should be very clear about what kind of answers will fall in the suit of this question and then pick the correct answers from the options, right? So one more minute and then we look at the answers. So you need to read. So see, for example, apoptosis, necrosis, autophagy, senescence. These are all related concepts, but then you will always find the key terms in the question, which will take you to the correct answer when the options are very similar to each other. But then there are words here like senescence, aging. These are all discussed in the respective sessions. So if you have to revise or if you are still unclear, you can follow the like the one which is specifically discussing these. Right. So 2 minutes over. So let me give you the answer. So the first one here, which talks about which process involves programmed cell death essential for development and homeostasis. As you know, this is apoptosis. The word programmed cell death was the most important because that program means that it is tightly regulated. And as I told you, they're essential for so many like important characteristics for the cell that they have to be regulated, right? Okay, and now let's just look at the other topics just to be clear about what those are talking about. So, for example, the second one is necrosis. Necrosis is basically uncontrolled cell death, right? So, cell death happens both by apoptosis and necrosis, but when it is programmed, when it's regulated, that's apoptosis. When it is uncontrolled, that's necrosis. And most often the non-necrosis is caused by injury or toxins or trauma. And the like the phenotype or when you look into necrosis, they will look like cell swelling, membrane rupture, inflammation. So remember, I told you that I was also talking in the previous slide. Inflammation is a characteristic of necrosis and not of apoptosis. In apoptosis, you see cell shrinkage, chromatin condensation, apoptotic body formation, blebbing, but not inflammation. Right. The other term with next option is autophagy. As the name suggests, auto means self. Phagy means to basically kill or eat. So that would translate to a cell survival mechanism in cells where they recycle damaged organism proteins by their own self. So basically, the idea here is to recycle. It is not necessarily cell death. It's mostly the organelles or proteins which are damaged, they are recycled. Right. And the last one is senescence. Senescence is a stage of permanent cell cycle arrest. There is no cell death there. It is cell cycle arrest. So the cell, they remain metabolically active. That means everything else is happening except for the division. So no cell division in the senescent cells. Okay, the next question. Sorry, just what is the primary characteristic of cellular senescence? As I mentioned, permanent cell cycle arrest, which I uh, also mentioned in the previous point. And what would trigger senescence in cell? When, so I told you, right, that cell division is very efficient process. It's supposed to be very efficiently done. So any perturbation or any damage or anything that can probably cause problems to the cell in later stages is prohibited. So if the cell sense any problem, it will stop the cell cycle progression and try to repair the damage. Right. For example, DNA damage or telomere shortening or any kind of stress like oxidative stress or nutrient stress and oncogene activation. So these will push the cells into cell cycle arrest, where they are basically enlarged, they remain as an enlarged cell. And then you have these remember we discussed about senescence associated secretory phenotype. So they will maintain the inflammatory factors that are basically telling that these are senescent cells. They are active, but they are not undergoing any division, right? And uncontrolled cell division, the other option, they are in cancer cells. So senescence are basically the opposite. Uncontrolled cell division happens in cancer cells. Enhanced metabolic activity is basically not a feature of senescent cells. They are metabolically active. The metabolism doesn't change. The only thing that changes in a senescent cells are the cell division, right? And then the increased telomere activity is again low. Telomerase activity is low in senescent cells because telomerase is basically again a characteristic of cancer cells, right? Okay, the third one, accumulation of which type of cellular damage is hallmark of aging. Aging is when basically the cell has undergone multiple round of cell divisions and there is progression in terms of the number of generation that the cell has aged. So all these problems can accumulate over a period of time, DNA mutation, protein misfolding, lipid peroxidation, and that all of them as hallmarks of aging. Remember, we also discussed 9 hallmarks of aging. So this is definitely, these are 3 of them. And which protein is a key regulator of apoptosis in intrinsic pathway? This we had discussed right now also. It's the PCL2, TNF alpha being a death like it, right? Okay, so the next set of questions, which of the following is not a characteristic of necrosis, the other way of cell death? A gain of function mutation in which type of gene can contribute to cancer development? This we had discussed in the previous problem solving sessions also. Which of the following is a common feature of cancer cells regarding energy production? And which of these is not a characteristic of apoptosis? So I will wait for 2 minutes and then I will discuss the answers. So basically, as you can see, um, even though apoptosis and necrosis are both of mode of cell death, there are some characteristic feature which are very different for both of them. So you should be able to, like if you remember one, the other becomes very easy to understand. So that's the whole idea here, that you can do such kind of exercise so that you can remember these topics. A gain of function, as I mentioned last time also, is basically a mutation that renders the functionality to that particular gene. Right. So let me give you the answer here. Okay, so the first one is, which of the following is not a characteristic of necrosis? And the answer is dependence on high ATP levels. That's because necrosis, like I mentioned, is, like it is caused by injury, toxins, infections, anything of that. It doesn't require energy, which is basically it doesn't uh, utilize actively ATP. So generally it occurs when there is depletion of ATP levels. So it doesn't require the need of ATP. For the other reasons, for example, membrane integrity loss, it's what leads to the cell death. Inflammation, as I told you, is a main feature, and cell lysis, of course, cell death. Right? So that's the first answer. Second one, a gain of function mutation in which type of gene contributes to cancer development. So proto-oncogene basically are those genes that can become cancerous, or that can lead to cancerous phenotype. So if there is a gain of function, it means the gene product becomes overactive or constitutively active because it has now gained that function. So a proto-oncogene, if it gains, if there is a mutation that leads to gain of the function being oncogene, that gets triggered and leads to cancer development. Which of the following is a common feature of cancer cells regarding energy production? It's Warburg effect, if you remember. So basically, Warburg effect is basically when the cancer cells produce energy through aerobic glycolysis. So what is aerobic glycolysis? They convert the glucose to lactate, and basically they do it even when the oxygen is available, right? If you remember from the Krebs cycle and etc. So normally cells would use oxidative phosphorylation, which is in the last step of your entire Krebs cycle. So you start with glycolysis, move to the TCA, and then to the etc. So oxidative phosphorylation, which is happening in the mitochondria, that's when normally cells would end up, and they will basically complete the entire procedure when oxygen is present, and so that they can produce ATP. But what the cancer cells do, they basically switch to glycolysis even if there is oxygen available. You don't basically like they don't really rely on oxygen anymore. So aerobic glycolysis, why do cells prefer to do that? Is cancer cells, because they would generate metabolic intermediates for biosynthesis, and the idea or the whole uh, thing here is that the cancer cell needs to grow quickly, so they would generate metabolic intermediate that they would use for producing different kind of biomolecules that they can use for proliferation of cells. So they want ATP production to be faster, even though if it is less efficient. So that's why you see this aerobic glycolysis in the cancer cells, right? And the last one, which of the following is not a characteristic of feature of apoptosis? Inflammation should have given you away. Introduction of inflammatory response due to cell lysis is not a characteristic of apoptosis. The last two questions for this part, before we begin with the week 7's topics. So what is the primary function of IAP, which are also the inhibitors of apoptosis proteins? So just thinking like this, what does apoptosis protein do? And if you inhibit those proteins, what can happen? And which factor is most important for angiogenesis in cancer cell? Angiogenesis means blood vessels. So which of these would lead to blood vessel growth or development of blood vessels in a cancerous cell? Okay, so these are two questions. Let me give the answers here quickly. Right. So the first one here is which basically which what are the primary function of IAPs? IAPs, as I mentioned, these are inhibitors of the apoptosis protein. So they inhibit caspase activity and suppress apoptosis. As simple as that, right? So basically, like as the name suggests, right, they are inhibitor of the apoptosis protein. So they block the apoptosis by inhibiting the apoptosis proteins. The proteins here being caspases. So these proteins inhibit the caspases. So they bind to these caspases like 7, 9, 3, and they inhibit their activity, right? And the next one is which of this is the responsible for angiogenesis? VEGF, as the name suggests, these are vascular endothelial growth factors. So growth factor that promotes the uh, development of particular, for example, here is vascular endothelial cells, which is really blood vessels. So VEGF is important for cancerous cells progression. Okay. So now let's move to this week's topics, which were about, like I said, regulation in cell cycle. Now here we will discuss topics which spans multiple topics that we have been discussing over the past week. For example, meiosis, mitosis, and the SMC proteins, which are basically the cohesin, condensin. We will look into them a bit more detail, because what the idea here is to understand them from a regulatory perspective. So these topics might seem repetitive, but then the idea here is to like look at them from the perspective of regulation. Okay, so let me first start with the SMC proteins. We have seen this slide already. We know SMC proteins are of three type: condensin, cohesin, and the SMC complex, right? Now, the first lecture of that week 7 basically talks about three concepts in great detail. First is about the these complexes in organizing chromosome. How are they organizing the chromosome? What is the role in the maintenance of the structure of chromosome? Then we talk about the chromosome loops, how they form. We know how they form loop. We have seen this in the previous slides also. And then we also want to understand what is the functionality of this loop and how do you study the loops? How do you see these kind of structures and how do you know the role of the essential proteins? Right. So just for your revision, so we remember, right, that you have in your S phase, your sister chromatids are formed because DNA replicates, and then the sister chromatids are formed. These are basically copy of each other, so they must stay together, and then they have to segregate only at the desired phase of cell cycle, which is the anaphase, when they have to move. So the other time of the cell cycle from S phase till M phase, they need to stay close to each other. They cannot just wander around in the cell. So that holding them together is done by cohesin, if you remember, right? And this is one of the SMCs. What is the other SMC doing, which is condensin? They are basically compacting the DNA into loops. That's when you see the loops, right? And that's because we remember a lot of importance of the groups. We'll also discuss this now. It's basically, so condensin, the molecule, it was basically helping to initiate or to like bring into effect chromosome condensation, which is an important part of mitosis, because this big chromosome has to compactly be arranged inside the nucleus, and then also be this has to stay like this till the cell cycle ends, right? So one uh, method that was discussed in great detail was the chromosome conformation capture, also called 3C, right? So let me try to explain to you what does this method do. So I will try to briefly explain these here, because the idea is that I have a lot of questions that can help you understand these topics also. The idea, so the thing is, I will try to make it explain in a simple terminology, but hopefully if you have it down, of course you can message, or you can solve the questions and try to grasp the topic better, right? So let me explain the concept of chromosome conformation capture and why do we use it. So this is a technique. This was also discussed in the lectures first lecture of the week 7. So it's this technique that we use to study the 3D organization of chromosome inside the nucleus. And when we talk about 3D, we are basically talking about the entire chromosome in its space. So that's when your condensin, your cohesin all comes into play, because that's we're talking about the arrangement of the particular chromosome or the DNA material inside the nucleus, right? So we, for understanding that, this technique, which is called chromosome conformation capture, is used, right? So what all do you see here? You basically can see how the DNA are close, which regions are physically close to each other, which are further apart in the uh, arrangement. So, so basically, you can imagine this is a DNA sequence, and you have one region here and one region which is after a certain gap. When you see them in the 3D space, or when you see them in the structure, these two regions are close by. So the DNA sequence doesn't necessarily follow how closely you are spaced in the region in the 3D space. And these kind of information has come from this technique, right? So there are several steps in this technique. And when we do that, we ultimately reach to the part where you basically take the conformation concept and go to the sequence and see how do these regions map, right? So first thing is, we all know we have this knowledge that DNA in the nucleus is folded, right? And looped. Loop we have already seen in the previous problem solving sessions, right? And because you have now folded the DNA and you have also looped the DNA, you will have that two DNA regions far apart in the chromosome can be physically close in the space. So you want to see if there is any particular rule governing that, right? Is there a rule which says that you have to be spaced to come closer to? Is there a rule governing how DNA extrusion takes place, right? So let's first look at the steps of this technique. So the first thing of a first step of this technique is to cross-link the DNA, right? So you have you treat the cells with a cross-linking chemical like formaldehyde. For example, you see here, right? So you lock the DNA segments together that are physically close inside the cell. So what it means is, this is consider this is the arrangement of the loops. And what you do is you cross-link them. So you bind them together, right? Then what you do is cut the DNA. So this chromatin is digested with any restriction enzyme. For example, here, as you can see, the names mentioned, DPN3, and you have other such restriction enzyme. So these enzymes, basically, as the name suggests, restriction enzyme, these are basically cleaving DNA. So they recognize a particular nucleotide sequence, for example, GAATTC, and they will cleave the DNA at a particular between or before a particular nucleotide, right? So you have EcoR1, you have DPN3, you have HIN3, you have BAMH3. All of them recognize their cutter sequence and then cleave the DNA at the respective location. So you have cross-linked the DNA, and then you use a restriction enzyme to cut these DNA into fragments, right? What you do next is ligation, as you see here. What are you ligating? So the fragments that were cross-linked together are ligated, which is basically it produces a chimeric DNA fragment made from two distant regions that were spatially close, right? So these were locked with each other. You cut the DNA, you have now ligated them together because they were close to each other, right? So you do this, this is what happens. Okay, so they are ligated with each other. Okay, so you have one region which is this, one region which is this. Now, what happens here is, if you see at this digestion here, this is basically, so this is one contiguous DNA molecule. You've cross-linked them, you've kept them at the place that they were. They were supposed to be close to each other, supposed to be close to each other. Okay. And then you cleave the DNA, make these two units. This is one unit, this is one unit. And then these unit, which is lying here, they're ligated with each other. So the loose ends are closed. So this is ligated here. This is ligated here, right? Now what do? Then you go for DNA sequencing. So from here, you shear the DNA, make it small, small beads, and then you basically anchor them, tether them. And then so what you do in this is basically, let me just read it out from here. Okay, and one point about why they should ligate, why she should bind to each other, is because of sticky ends. So I don't know, I'll try to show you here. Yeah, so basically, this is your DNA sequence. And when you do restriction and ligation, first is restriction. So you make some sticky ends. So let's say it cleaves here. Complementarity will also lead to cleavage here. So you will have a sequence like this. And then here you will have a sequence like this, right? This ends and these ends are called sticky ends, right? Means, so for example, you have G, A, T, T, C. These are palindrome sequence, right? So they have to be G, A, T, T, C. So they cleave here and they cleave here. So this part and this part look similar. They are sticky ends. So that's why when they cleave it, if you allow them to come together, they will again form, they will ligate with each other. That's what is the idea of ligation, right? So that's what is written here, that sticky ends are created by this enzyme, which are ligated to create loops like this. And then these linking pieces that are far away are now in one particular conformation. This DNA is then sheared into small, small pieces, and then you pull them out or you tether them with your streptavidin uh, anchors or streptavidin molecules, and then you do DNA sequencing to get the sequence of the DNA. And what will the DNA sequence tell you? That which regions are interacting with each other. So, and what does it give you? It gives you a contact map. It basically tells you the nearby regions which are part of the DNA structure. And when you look at the signal, how far they are and which of these are coming close to each other. And then you have some, like you can identify regions on the DNA sequence which are part of chromatin loops and which are part of TATs, topologically associating domains. So both the loops come together. So those are topologically associated. So the TATs. So you can identify these regions using this method. And that's when you realize that chromosomes are not randomly folded. They have a, like they have a mannerism how the domain, how they form the domain, which domains come close to each other and then form the loop. And because this happens, they influence gene regulation and chromosome function because they make these loops. We have also done this last time. They make accessibility to the of the particular region of DNA to the transcription factors or to the polymerases to carry out translation, transcription, different kind of uh, processes in the cell, right? So how do particular regions only undergo cells regulation or translation? That's because these are the regions that come close to each other and they are accessible by the other molecules, right? So that's was the idea here. So this is what the loop looks like. This is how they are formed. So this just we used to understand which loops are these, which regions come close to each other. These rings we had already discussed in the previous class, right? And that's the whole idea in the first lecture of this week, to understand how DNA loop is formed. So we know it's not random, but what is the exact functionality of loop, that is still not clear. So let's try to do more questions so that we get an idea of these topics much better. So again, I will wait for 2 minutes in each slide and then move ahead to discuss the answer. So the first question is, what is the primary role of cohesin complex during cell cycle? Which family of proteins organizes higher order chromosome structure? Easy question. Condensin complexes primarily function to do what? What is the use of condensin? And why is chromosome condensation at all necessary during mitosis? Why is it so important for the cells? So cohesin, condensin, like I mentioned last time also, the names of the proteins suggest the activity they do. Cohesin means keep them cohesive. Condensin is basically to condense any particular molecule. So that should tell you what kind of functionality they have. Yeah, so there are 4 questions. So I will wait for a minute more and then we can discuss the answers. Anyone else? Any other answers? Any other guesses? Okay. So yeah. So maybe of 30 seconds more for anyone else to try and attempt. Then we can look at the answers. Okay. So let me give the answer. Sorry. Yeah. So the first answer for the first question, the first is that what is the primary role of cohesin during cohesin complex during cell cycle? And as I mentioned, that the role is to keep the sister chromatids together. So this basically tethers the sister chromatids during the S phase till their separation later in the M phase. Okay, next one, which family of protein complex is responsible for higher order chromosome organization? We already have discussed this, SMC proteins, who basically organize the chromosomes and they are found both in prokaryotes and eukaryotes. Just for clarification, SMC's full form is structural maintenance of chromosomes. So the name itself suggests that they are responsible for how the chromosomes are organized and maintained. Right. Third one says condensin complex primary function condensin. So it refers to compacting the chromosomes during mitosis. And how is that compaction done? That compaction is done by the DNA loop that we had just discussed so far. Okay. And the next one is, what is the main purpose of chromosome condensation during mitosis? Why do you at all want to do this? That's because you want to prevent chromosome entanglement and allow the movement of this DNA into the new cell which forms after the cell cycle. Okay. Right. So the answer, as you would have seen, is B, 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 and C. Right. Okay. Moving to the next set of questions. Just a second. Yes. I hope it's clear to you. So chromosome conformation capture, which we have discussed right now, what is the main goal of using this technique? And then when you get a contact met from this 3C, what is the nearby nucleosome interaction? Why does it take place? How does it take place? And interphase chromosome loops. So what is interphase? If you are able to imagine, then you will know how long are the loops for, or interphase chromosome loops detected in metazoas are how long. And then which protein position chromatin loops in mammalian cells. So just what I told, which proteins help in that conformation. Any attempt, any answers? Okay, a minute more and then I can discuss the answers with you. If you have any guesses, you please take your guesses. I mean, the idea here is to help you think about this topic. This topic might look very overwhelming if you haven't read. So that's all right. That's the whole agenda for this session. But if you have read or if you want to revise, you can just take a guess and you can see the answers and think if you were thinking in the correct direction. So a minute more and then we discuss the answers. Right. So let me give you the answers for this. Okay. So the first one here is chromosome conformation, or the 3C technique is used to study chromosome interactions and spatial organization. That's what the idea is, right? Which regions of DNA are interacting and how they are organized in their 3D space. Next one, in a 3D contact map, interactions between nearby nucleosomes will appear along the diagonal. Okay, so a simple idea here. You have, so remember you go back to the DNA sequence and you find out, okay, this region and this region are close to each other. So you realize, so let me just make it simpler. This is a DNA sequence. I put it in 3 parts, 1, 2, 3. In the loop, you have 1 and 3 coming closer to each other. So the part 2 in the DNA is in the, so it's not in the loop. Region one forms a loop, region three forms a loop, right? So you know that region one and region three contact with each other. So based on these information along the chromosome, along the DNA sequence, we'll make a map, okay, and we'll plot them according to their interaction or according to their distance, right? So you have region one, region three, they are interacting, so they will be along the diagonal. So you will see. So if you have a plot like this, let me try to make it here and show it to you. Right. So this is generally the kind of plot that we will make for interaction map that we'll get from here. And this is a diagonal, very bad diagonal, but the idea here is that along this diagonal, you will see the contact points. So you will see region 3 here, region 1 here, region 2 here, region 3 here. So these will tell that they are in close conformation with each other. The others are spaced out according to their distances, right? So that's why whichever is nearby nucleosome interacting region, they will appear along the diagonal, right? So next one, which is basically the loops which are detected in metazoans, they are basically around 550 to 1000 kb long. So they are very long, right? And lastly, which proteins help in this loop formation? That's CTCF formation in mammalian cells. So CTCF is basically the DNA binding protein, and this binding of the protein or the binding on the molecule, the DNA, basically act as the boundary element where the loop positioning start. Remember, we had done these CTCF loops, rings, and we had made them. We saw how they loop out. So CTCF is a protein that helps in loop formation. Okay. And then I have one more set of question for this part, which is basically SMC complexes were discovered to, what is this process called? Evidence of loops. And how do you capture this is also very easy. Which protein limits loop expansion along chromosomes? Limits mean, how do you know that, how do you determine the edges? And chromosome looping may help. This is the importance of looping. Why do you need looping? So a minute or two, and then we discuss the answers. A minute more, and then we discuss the answers. All right, let me give the answers to you and discuss the answers as well. Okay. So the first one was easy, right? So the first one says, what is the property of SMC complex that were discovered by experiments? That was actively extrusion of DNA loops, right? So the extrusion is basically pulling them out of that particular region. Which experiment led to this evidence? 3C, or chromosome conformation capture. Now, which protein loop limits the loop expansion? It's the WAPL, or so basically these protein limit the loop extension. They define the region along with CTCF, and a proposed so what's the function of chromosome looping? It's basically a regulation of DNA repair accuracy. So basically looping can guide the homologous recombination repair by bringing the correct repeat sequences together. So they are important for genomic stability, and they bring, so like I said, they can bring regions for homologous recombination, for structural maintenance. So that's why looping is very important, right? Okay, so that was the first topic for the week 7. Let's move to the next part. Since I already spoke about recombination here, let's move to understand how recombination takes place and what exactly are the topics that were discussed in the second lecture of week 7. And we already know a bit about recombination. What does recombination mean? What we have a fair idea about what would recombination lead to in a cell cycle, right? I spoke about diversity. I spoke about genetic the variation that we see in nature comes from the fact that there is homologous recombination. There is crossing over of genomic information taking place during the cell cycle in the meiosis stage. So you can see here that, for example, this is your homologous chromosome, sister chromatids with their sister chromatids. So this thing, remember, this and this are called sister chromatids. Sister chromatids become or they come up when there is S phase, that is DNA replication. Otherwise, you have this one strand A and B from parental, both the parents. So you have one copy from mother, one copy from father, and both these copies duplicate during S phase. And this is how it looks under cell cycle when you see under microscope. So you have this and this together, they are called homologous chromosome. One of them is called chromosome. Now, in mitosis, there is no crossing over. They just simply split at this point and they move into opposite poles. But then there is meiosis. There is this step in the prophase of meiosis 1 where you have genomic information exchange. So let's say this is a parental copy from the father's side and mother's side, and you can see the genomic content in the letters that are mentioned here. So capital A, capital B, capital A, capital B. These are exactly identical because they are sister copies. So you have a, small b, and the sister copies. Now, when you look into the chromosome crossover, what we see here is that this part of the DNA and this chromosome and this part of the chromosome exchange material. So after chromosome, there is a chimera, or there is exchange of information, right? So you have this A here, which takes away information from B, small b, so capital A, small b, and then you have one region which has small a and a capital B. So these kind of crossover takes place. Now these two chromatids, remember, even in the meiosis at the end, all of this will split. Okay, so this will go to one pole in one cell, this will go to one cell, this will go to one cell, and this will go to one cell. So when you have, so pen, when you have this particular chromatid going to a new cell, this is a non-recombinant. Why? Because this is exactly like the parent chromosome. When you have this chromosome into the cell, that cell is also non-recombinant. Why? Because this looks like this. But when you have this one here, this is a recombinant chromatid because this one has information from both blue and red, which is not there in the parental chromosome. And similarly, this one here is a recombinant because this also has combination of genetic material. And because there is a change in the information across generation, this is the variation that we talk about when we are saying that there is standing genetic diversity in the nature, right? But this crossover or the exchange is also highly regulated. And that's also something that was discussed in the, like in the lecture in the week 7, second lecture, right? Okay, so let me try to explain to you how does this takes place. So first and foremost thing is for your, this is also a highly regulated process. So you have these DSBs, okay, and which is double stranded breaks that's what guides the recombination. Okay, now before I talk to you about this figure here, remember from the previous lectures or previous sessions that your meiotic prophase 1 is separated into 5 stages: leptotin, zygotin, packaging, diplotin, right? And then dikinesis, right? So now what you see here is when you are in the, just for a revision, your leptotin is when you have chromosomes begin condensation, okay? And then you have one another enzyme that comes into play, which is called SPO11, which induces double-stranded DNA breaks. So you see it here. Okay, so your chromosomes have compacted, and then you have this nick here. Nick means basically a cut, and your SPO11 and the likes of those enzymes, they form something called DSP, double-stranded DNA breaks. Okay, so there are breaks here. Okay, so you have breaks here. So that is happening in the first stage of prophase 1 of meiosis 1. Okay. Next step after leptotin is zygotin. When you have the homologous chromosome, they come and start pairing with each other. And then there is formation of something called synaptonemal complex. And that's when your recombination will begin. So the next step here is, so you have, sorry, you have these homologs coming and pairing with each other. Okay. Now, next step, which is basically packaging, when your step has completed. Okay, this is just an overview first. So your packaging synapses, so the crossover has completed, and they have formed this link. Remember from the last slide, maybe it will be clear in the last slide. This one here. Okay, so this is formed. Okay, and in diplotin, you have homologs now started separating. So this junction that was formed, sorry, is broken off and they become chromosomes, separate chromosomes again. So this all is taking place in the prophase 1. Okay, so what begins the entire process is SPO11 and other enzymes, which basically will make these double-stranded breaks. Why do you need the double-stranded breaks? Because you need this gap in your DNA here to begin the invasion of other strand. So let's say you have a strand break here and a here, and you, so the DNA would like to form a pair once again, right? DNA would not like to be in the loose end, so it will start forming or close the gap. Sometimes or during recombination, what it will do, it will invade the other strand. Instead of going to its native strand, it will go to the other copy. So this is strand invasion, and it can only happen if there is a double-stranded break, right? So this is intersister invasion, and if so, you see all of these here in the crossovers, right? So, right. So first thing is breakage. Next point is resection. Now, after your break is formed, the broken DNA ends are resected, and they are basically these DNA tails that are formed. And so Three sections mean they are loose end, right? So they will start looking for their homologous sequences. Next, as I mentioned, it will be strand invasion. So you have this DNA invading the homologous chromosome. Like I said, there are two possibilities. Okay, first is interhomolog invasion. This is exactly what should happen. But then can also be case, which would have imagined while I was talking, that is intercystal invasion. So this is basically the alternative repair pathway. Okay, so intersister. So you go back to your sister's copy. I mean, you go back to your copy and you form a pair there. Because remember, they are double-stranded breaks. So here, as you can see, it can also go here and here, right? So that is not something that will give rise to diversity because they are copies of each other. Only where it invades this other strand, that is what you want. So this thing is not uh, what generates diversity. This will generate diversity. Okay, and you can see the diversity already here. Okay, right. So now after your invasion has taken place, once the invasion is complete, what you do is the second DNA end is also captured and it's called something called holiday junction. So there's a junction as you can see here. This one invading here. So what I'll tell you, explain it better. So this thing comes and binds here or starts invading this strand. This strand will move out. And as this moves out, this comes in vicinity to the other end and it is pulled by the other strand to come and bind to it. So you have uh, these junctions here, right? These are called holiday junctions in the on the name of the scientist who discovered it, right? So you have these junctions here, right? And you have several proteins that will be here maintaining that structure. Okay, now what happens after this? Again, there are two type of crossovers. One is class 1 crossover, one is class 2 crossover. One crossover requires the ZMM. Okay, so basically here you have this double holiday junction that I just show, and the junctions are resolved and this produces crossover. Basically, what I explained to you that. So yeah, just move from here. So you let me just wipe. Let me remove this. Yeah. So you have. Okay, now see this what will happen. Okay, this is class 1 crossover. So you have this interhomolog strand invasion. You have this one being pulled to the next strand. A blue, red, blue, red, and red. Okay, blue, this blue, follow this blue. This will have a particular red and it will then continue to be the red. The other one will have part of its red and then parts of the blue and then it will be. So what I mean here is that you have this double junction here. Now look at this end product and look at this end product. They are very different. Why? Because in this you have the exchange of material at two locations. One is here and one is here, right? Here on the other hand, you have the exchange at one junction and then you have the other part. So if it's blue, then it's red. If it's blue, then it's red, and vice versa. Here, if you have blue, you have red and then a part of blue and then again a red, and the other strand is blue and red. So one of the strand has multiple components. That's the kind of crossover depending on at which junction the exchange took place, at both or only at one. So that's the whole idea. And if the crossover took place only on one of the sister chromatids pair, so from here and here, then it will give you two kind of products, two which are crossover products and the ones which are non-crossover products. Okay, so there are multiple outcomes that you can have from crossing over, but the idea is when you have these are the steps that are part of your recombination, and that can give rise to your diversity depending on which of this fate is met at the end for the cells which are dividing. Okay, all right. Now coming to the like the regulation, all the steps that I just mentioned has so many proteins that come and take part into it. So remember, these are the steps of meiosis one. What I just spoke to you about is just this part of your meiotic prophase one. You have meiosis two, in prophase one, you have several steps. So all of these, that's why I was taking those gaps because I didn't want to name it at that point. So at every step, you have several enzyme proteins, for example, IME1, IME2, that are transcription factors or kinases that will drive the cell for completion of that particular cell cycle phase. CDKs we already are aware of, but then outside CDKs also we have several other proteins which are important that hopefully will, as it comes up in the questions, it will be helpful for you to remember. Okay, so let's begin with the question so that we try to grasp the topic better. So the first one is, which protein induces meiotic DNA double-stranded breaks? And meiotic double-stranded breaks preferentially occur in which part? Sorry, what prevents, just a second, I will fix this up. Sorry, I fixed that part. Yeah. What prevents multiple double-stranded breaks? How does it stop? And which histone modification marks DSB hotspots? So we'll also see what do these words mean. I'll wait for 2 minutes for you to grasp the topic and then we can move. Okay, a minute more and then I'll explain the answers. Okay, let me give the answers. Right. So the first question, which protein induces meiotic DNA double-stranded breaks during homologous? That's we have discussed. That's right. That's SPO11. This is a topoisomerase-like protein. Remember, topoisomerases are proteins that are responsible for maintaining the structure, especially in the cell cycle, where they are basically maintaining the structural integrity of the DNAs, where the DNA have started looping out in S phase and where the replication junction is forming, right? Similarly here, this is a topoisomerase-like protein, which basically induces the nicks in the DNA strands, okay? Next was, during meiosis, double-stranded breaks occur preferentially in the intergenic promoter regions. So these are basically in the central part of the DNA sequence. You generally have less of such recombination in the telomeres and near centromeres. Centromeres, as we know, are very important for kinetochores, for attachment to spindle fibers, the cohesins and the condensins. So except for these regions, which are very important for otherwise cell cycle, the regions which actually promote to genetic diversity are these intergenic promoter regions, which are where you have more of this GSB frequency. Next one, which phenomena prevents two DNA double-stranded breaks from forming close together? So that's the double-stranded break interference. That means if you have one DSB, one breakpoint, the other breakpoint is suppressed in the region which is close by. So let's go back to the figure here. If you have a DSP here, another DSP here is suppressed. The DSP could be somewhere here. So what does it look like in the entire sequence? You have one chromosome exchange here and a chromosome exchange here. So you have diversity, but you still maintain the like the important functionality of other parts of the region of DNA. So for ensuring that it does not generate such diversity that everything is changed and the major protein function also changes, the DSBs are uh, spaced out. So they are in a controlled manner. The homologous chromosome also takes place in a controlled manner, right? So that's why intergenic promoter region. And the last one, which hosts histone modification, marks the meiotic hotspot. So where are these hotspots present? How does a region become the target for DSB when there is this presence of H3 K4 methylation? What is H3? Histone 3, K4 being lysine 4. So methylation, which is the epigenetic marker, and the presence of these methylation patterns marks it for DSP formation, right? That's why if you have epigenetic modification, it might also lead to the loss of such associated features. For example, if you remove the methyl transferase activity, if there is decrease in the methylation pattern, then your DSPs won't form. And if your DSPs don't form, you don't have that many recombination taking place. And that's why you will not have that many variation, right? Okay, next set of questions. So the chromatin remodeling helps SPO11 bind. So SPO11 is basically the DSP. So what does remodeling have to do with SPO11? What is obligate crossover? What is an obligate crossover? Crossover interference, which you would have just got an idea from. What does it ensure? An entry into meiosis in budding yeast. So budding yeast, we will do it in. So we'll discuss yeast in a detail after in the last topic. So, but just an idea, how do you have budding yeast meiosis taking place? A minute more, and then we discuss the answers. Okay, let me give the answers, discuss them. Right. So let's try to understand the correct answers here. The first one says chromatin remodeling factors. For chromatin remodeling, how does it assist SPO11? It assists them by giving access to the DNA. Now, SPO11 is basically supposed to make these DSBs, right? So you have a pair of homologous chromosomes and you want to initiate the breaks, but you can only do if you're able to reach the DNA. So for reaching the DNA, you need regions, accessibility regions. Those are given by chromatin remodeling. So any the proteins which are responsible for remodeling of chromatin, which is basically opening up the chromosome, making it a bit looser. So those proteins will first allow the DNA to be accessible to other factors. Remember, we have discussed this in looping and everything. And then the regions which are accessible are then encountered by the SPO levels, right? Okay, next one, what is meant by an obligate crossover during meiosis? It means that there is at least one crossover per homolog pair. So you have, so the idea here is that every homolog pair that you see, there should be at least one crossover. I said there can be multiple DSPs, but obligate means at least, if not multiple, one has to be there per cell cycle, right? Why? So that it ensures the proper segregation at the later, because there will be chiasmatic formation, it will break over. So as the machinery is built, that at least there has to be one crossover. That's obligate crossover. Obligate means has to happen. Okay, next one, crossover interference, which we just discussed, right? One DSP, so the other DSP will not be closed, that will be suppressed. And that's to make sure that the crossover regions are spaced apart along the chromosomes, right? Even distribution of these recombinant events. And the last one here was in budding yeast, what ensures the entry into meiosis? That's nutrient deprivation or nutrient limitation. And basically, these limiting factors will activate meiotic regulators in yeast, and then that will initiate the entire meiosis process. Because otherwise, they reproduce by fission, right? Budding. So, for them to actually divide, they need specific cue or keys from the surrounding, right? And we'll discuss that in the last part. Okay, now the last set of questions for this part. Again, IME. I just said in yeast also you have IME, which is activated under nutrient deprivation. So what would IME do? How will you make sure that your meiosis starts? Which checkpoint monitors recombination completion? So how do you, how, what ensures that recombination is finished and now we have to proceed to the next step of cell cycle? Which enzyme cleaves this cohesin, Rec-8 cohesin during meiosis, and why do you need to cleave it at all? And why is centromeric cohesin protected in meiosis? So this is very important and very easy also to remember, not that difficult. Right, a minute more and then we discuss the answers. Okay, so I give the answers. Right. So the first one says IME promotes the entry into meiosis by degrading S-phase CDK inhibitors, right? Remember when we first began this entire course, we studied about cell cycle and we knew that at every stage of cell cycle, you have checkpoints. You have inhibitors that are there to prevent early transition into the next phase, right? You only enter from S phase to G1 phase when there is completion of that phase and everything that has to happen in that phase. So you have these inhibitors that control the CDKs. Remember, cyclin dependent kinases. So you have CDK inhibitors which stop the pairing of the CDK and the cyclin and then the transition into next phase. So if you want to now transition into next phase, you basically just degrade those inhibitors, right? So IMA will basically degrade the inhibitors of S phase, and that will promote the entry of the cell into the S phase and continue with the meiosis here. Okay. Next one, which checkpoint, sorry, just a second. Yeah, which checkpoint monitors recombination completion? In other words, which one checks that whether the double-stranded breaks that were formed have been repaired? That's packaging checkpoint, just like cell cycle. At every subsequent stage, you have a checkpoint. So you have a packaging checkpoint here, which will basically arrest the cells in G2, right? After G1, you have S, then you have S2, sorry, G2. So it will arrest the cell in G2 if recombinant intermediates or DSBs are not repaired. Okay. So next, G2 means the stage that was, because prior to your M phase, you have G2. So if you don't progress through your M phase, you are basically arrested in G2. Right. Next one, which enzyme cleaves the cohesin subunit RECATE, or as mentioned here, RECATE cohesin during meiosis. So that separase, again, as I mentioned, the name of the protein gives away that function. Cohesin holds these sister chromatids together. So the separase is basically to separate these chromatids. So separase is basically breaking apart these cohesin so that your sister chromatids can move to the separate poles. And the last one is during meiosis 1, why is centromeric region protected? As I told you, centromere is a region which has kinetochore where the mitotic spindle comes and binds to. So if you basically, there is any problem to the centromere part or centromeric cohesin for that matter, then the entire step of the spindle fiber formation and these sister chromatids moving to opposite pole gets disrupted. So you basically do this to allow the homologs to separate, but the sister chromatids remain together, right? Okay. Now, it has already told you about, so that's why homolog separation. Remember the concept of sister chromatids and homolog chromosome, homologous chromosome. In meiosis 1, homologous chromosomes separate. In meiosis 2, your sister chromatids separate. Meiosis 2 is similar to mitosis. Okay, right. Okay, now that we already spoke about mitotic spindle. The third lecture for this week was about mitotic spindle only. We already did about the regulation and the formation of mitotic spindle, but as I said, this topic might look similar. We have been discussing them so far, but the idea here is to make you understand that everything that happens in cell is highly regulated, right? So even if they were discussed in bits and parts in the previous uh, lectures, they have been amalgamated here in this one lecture to again reinforce the idea that they are highly regulated. So we already know about mitotic spindle. We have discussed this so far, right? So I will just go over the important topics so that you get an idea about what exactly was discussed in this lecture. So mitotic spindle, as you know, is a bipolar. Anyway, this is the last topic that we had studied, right? So these are the bipolar microtubule structure. They segregate the sister chromatids. You have the centrosomes, which are the microtubule organizing center. We looked into their particular structure. They had centrioles and the pericentriolar matrix PCMs, and they also duplicate during S phase because you need two of these to go to the opposite poles, and then they will basically you have the gamma tubulin ring complex. We did all of this. So I'm just trying to just tell you the keywords which is important to you. Then we looked into the three type of microtubules where kinetochores were there, interpolar were there, and astrals were there. And the idea was that they have alpha tubulin and beta tubulin heterodimers, which form the microtubule. And they are also basically uh, growing, replicating, going to the opposite pole, binding to the chromatids and pulling them apart, right? And one important thing here is that the kinetochore concept is basically these are the regions on the centromere which connects the chromosome to the spindle microtubules, and that's why in the previous question, last question, you protect the centromere from any separase activities, right? You have cohesin here in the centromere, okay? And a few other things that we had read was bioorientation. So your sister chromatids must attach to the opposite spindle pores. poles, right? So you have one sister chromatid binding to only one spindle fiber, and that's because when you are in the metaphase plate, one of the sister chromatid binds to this side and the other sister chromatid binds to this side of the cell so that they can move to the opposite poles, right? So this is by orientation of the spindle fibers so that they can create the tension. And then one important enzyme that we had read was aurora B kinase for the like for the orientation of these spindle fibers. Okay. And then, of course, you had checkpoints. And remember the about the anaphase trigger here. So your sister chromatid separates in the anaphase stage when you have the APC and CDC20 activation, which is ubiquitin and proteasome, where basically you need to degrade securin. Securin is the protein. Just a second. So securin is a protein that keeps everything in place. Separase is what moves them apart, right? So when you want to separate them in this towards the end of anaphase, you have securin degradation, separase activation, and the put the sister chromatids away. So this we have all discussed all of this. I just wanted to give you an idea about what did we discuss and go over the questions just so that you can revise this concept even in this part of the problem solving session. So we'll do this quickly. Which structure segregates chromosome during mitosis or meiosis for that matter? Remember the contractile ring that we had discussed about. So what does it contain? And the primary MTOC, which is microtubule organizing center, which is a primary center in cells, animal cells. And the centrioles, we had discussed the orientation and the composition of centrioles. So this question basically talks about which of these is the correct answers for the organization or the components of MTOC. A minute more and then we discuss the answers. These are questions that we have, I mean, these topic in itself we have discussed over the past sessions. So like I said, we have done it bits and pieces. So the idea here was to pull all of them together. So it might sound repetitive, but then the idea is to just put them all in one place and reiterate it. Okay, I'll give the answer so that we move to the next part. Right. So the first answer here, or the first question's answer here is um, which cytoskeletal structure is responsible for segregation of chromosome during mitosis? That is mitotic spindle because they are the one that forms in the opposite orientation, connects or binds to the chromosome or the sister chromatids and pull them apart. Next one, the contractile ring responsible for the cytokinesis is composed of actin and myosin because these are the molecules which are dynamic. So these dynamic proteins can move from one region to the opposite poles and then bind to the chromosomes. So the movement or the dynamics of the microtubule or the spindle fiber is because of the presence of actin and myosin. The MTOC or the primary microtubule organizing center in animal cells is centrosome that we discussed extensively in the last problem solving session. And centrioles in the centrosomes are characterized by 9 triplet microtubules, which also is something that we discussed in the last session where we looked into why we have this radial symmetry in the mitotic spindle. Okay, next set of questions. Which complex nucleates microtubule? We'll see what it means. How many protofilaments form a microtubule? Microtubules are polymers of which of these proteins and which nucleotide regulates beta tubulin dynamics? We'll wait a minute or two for your answers, and then we can discuss the answers. Okay, a minute more, and then we look into the answers. Okay, let me give the answers. Okay, so the first question was which complex nucleates microtubule assembly? So what basically ensures that the units of microtubules are added? That's gamma tubulin ring complex. Okay, so these gamma tubulin complex is acts as a template for the nucleation. So the microtubules come together because of this factor. A microtubule is composed of how many protofilaments? 13. Okay, so there is a central, it's a cylindrical structure which in the it also has a hollow lumen. We discussed this and we looked into the structure in the previous session. Microtubules are polymers of which proteins? These are tubulin heterodimers. So heterodimers mean two different type of component, not exact copies of each other. And what is that? Those are alpha tubulin and beta tubulin. So your alpha tubulin and a beta tubulin, they form the filaments, while your gamma tubulin initiates their nucleation or basically helps in the complex coming together. Okay, and then the last one is which nucleotide regulates microtubule stability by binding to beta tubulin. If you remember about the GTP hydrolysis and GTP hydrolysis, so if you have GTP T triphosphate, GTP bound beta tubulin, it stabilizes the microtubule. But if you have hydrolysis to GDP, it promotes the destabilization. It promotes the recycling or the breakdown. Right. So for the stability, you need GTP. All right. Okay, and the last set of questions before we move to the last topic. So if you have loss of GTP, exactly what I mentioned, what does happen? Transition from growth to shrinkage is called what? Which structure connects microtubules to chromosomes? That's the whole idea. And which kinase monitors the incorrect chromosome? So if there is incorrect chromosome orientation, which of these comes into play? Not a difficult topic because it's fairly easy to understand. Just by the name also, you should be able to take a guess of what could be the answer. Okay, I will wait a minute more and see if anyone else has to answer. Okay, I will give the answers now. Right. So as mentioned already, loss of GPT, basically GTP cap at the end of microtubule, if you remember, in the details. It basically leads to rapid depolymerization because it becomes GTP and everything is then starting to disentangle this or to move away from each other. So if you have GTP to GTP, like breakdown, then it causes the protofilaments to curve and the microtubules to disassemble. Okay, now the transition from microtubule growth to shrinkage, if you remember the entire cycle, right, of how the microtubule duplication and growth takes place, the step of growth to shrinkage is called catastrophic, right? Because in general, microtubules dynamic instability, which is basically why they can move into different forms, includes the growth. We discussed this. Catastrophe is a shrinkage, and then you have rescue events. So anything that has to deal with improper organization is basically catastrophic. The next one is which protein complex connects spindle microtubules to chromosome. We have discussed this multiple times. Kinetochores, which are assembled at the centromere. And the last one, which kinase sense tension at kinetochore during chromosome alignment. It's Aurora B kinase, right? We discussed this. So this enzyme, this kinase, will detect any incorrect attachment and then phosphorylate, because that's what kinases do. They phosphorylate the kinetochore proteins, and this is done to destabilize the attachment, and the microtubules will go up because it is not correctly arranged. So that's the whole idea, right? Okay. And now we move to the last topic, which is basically discussion of a particular cell type. So today we'll do yeast, and in the next session, which will be the last session for this course, we will do plant cells. Okay, we'll see why is yeast important. Okay, so yeast is basically Saccharomyces cerevisiae, and we will see the features of yeast that makes it amenable to study, not only the features of yeast in itself, but as a model organism. A model organism is any organism that you can study the processes and then take it for higher organism, right? So what makes yeast a good model organism? What processes, what uh, features of yeast have already been studied, and how have they given us an insight into the other processes or similar processes in other organisms, right? So this on your slide here, it just shows how does a yeast look like under microscope and what is the cell structure of yeast, right? Okay, so let's just discuss again. So we have yeast, and we are particularly discussing the budding yeast here, which is Saccharomyces cerevisiae. So these are fungus. They're eukaryotic fungus, and if you have to mention some common features, you commonly call them as baker's yeast because you use them in baking. Cells are very, the size of the cell is 5 micrometer. And budding yeast comes from the term budding, which basically is a mode of their reproduction that we will see in the other slides. Okay, so because they are eukaryotes, they have nucleus, they have mitochondria, they have the plasma membrane and the cell wall also. And now if you look into the cell wall, that also is very, that has been studied to a greater extent. So in the cell wall, you will see there are two layers. One is the inner layer, and then there you have an outer layer. So the inner layer has all the glucans and chitins, and the outer layer has manoproteins, mannose, and so like a proteins which are mannose subunits. So this cell wall, as like other cells, they maintain the cell shape, they will protect the cell during growth. And these are the, because I told them they are fungus, right? So these are the targets for antifungal drugs, right? Because that's helping the cell to retain its integrity. If the drug targets the cell wall, the integrity of the yeast is disintegrated, and then you basically can lead to the death, or you can basically remove the source of infection, right? Okay, now, like I mentioned, it's a budding yeast. So let me try to help you understand what budding we are talking about. So this is basically also reflecting on the cell cycle of yeast, on their mating, on their budding, right? So we'll do that in extent. First, let me just tell you overview. So yeast, they divide asymmetrically, as you can see from here. Just a second. So as you can see from this part that the cells are not uniformly distributed, okay? So they will have two and two and two, and we'll look at all of these conditions. Just the first thing to remember is they divide asymmetrically, and there are several steps in the cell division. But the important steps, things to remember is they also have cytokinesis as with other cell cycle that we have read. And then there is a polarized growth, and there are in cell cycles of yeast, you have remnants of previous division. You have marks of previous divisions. Okay. And like I said, it's also reflecting upon the yeast mating. So just overview first that there are two types of mating types in these yeast, which are haploid here, as you can see. Okay. So these haploid yeast have two mating types, MAT A and MAT alpha. Okay. And the mating, which is what would lead to 2N, is basically under trigger. There are some triggers that will allow the cells, yeast cells to mate. Mating has again two type, MAT alpha, MAT A. But once the pheromones are there or the triggers are there, the yeast cells which are there will have a G1 and will be in G1 arrest. There is something called smooth formation, and then the cells will fuse and they will form diploid cells. Okay, and then this diploid cells, which is here, it will undergo formation into 4 haploid spores. We will see why. Okay, so let me begin with the cell cycle in a bit more detail so that it helps you understand what I'm talking about here. Right. So in general, they reproduce by budding. Budding is just offshoot. So you have your cell body, you have offshoot, and it just forms. So they're basically exact copies, right? But so when it is happening, that's why you have asymmetrical cell division, right? Because it's just offshoot and it's forming. It also has its G1, G, G2, SM, all those phases that we have read about. But um, the importance here is to understand the budding here and all the steps that I just showed you, that it's not just an offshoot and it grows away. There is a systematic way of how these steps takes place, right? So let's begin. The first thing is the mother cell produces a small bud that grows and eventually separates as a daughter cell, right? So you see this here, daughter cell separation. So this is budding basically. I don't know why the cursor is not moving properly, but let me try and show from here. So this is budding. Sorry. This one is budding. So you have a mother cell and a bud, and the bud is separated and the mother cell is separated, right? So in this budding cell, so when you have these buds forming, there is, remember, just try to imagine that there is a cell wall and it's a bud formation and it separates out. So every time the budding takes place, there's a scar that remains behind, and that's a mark that the cells have undergone division. Okay, so it's called bud scars. Okay, so here you have the bud site selection, which happens in the late G1 phase. Okay, so the cell will determine where this new bud will form, and this process will be highly controlled. And then you have this axial budding or the bipolar budding. So you can have bipolar or you can just have one bud forming. And there is an enzyme or there is a small protein called CDC42 which helps in this. Okay, so then a bud is formed. There is wherever that protein is accumulating more, that's the site of bud formation. So you have this bud formation. Now what do you need? You need this to fall as a separate cell, right? So there will be a formation of septin ring. Okay, so at the bud neck joint, it will form a ring, and it will initiate the process of cytokinesis, as we know. Right, so it will form kinases, and then you have actin filaments and cables which come and disentangle this, and then with the help of these actin and microtubule and all the uh, cytoskeletal proteins and filaments, these bud will come off from the mother cell. So that will relate to this formation of budding and their mother cells after their reproduction, right? But then there is also nuclear division and cytokinesis that happens during mitosis that we already know of, and that takes place during the mitotic spindle, because remember all of these I told about G1 phase. There will be S phase, duplication, and system. That every process that we have read will be taking place here also. The only thing that here you see here is this part here is what I have discussed so far. We also have this part here, right? This mating here. And then it also comes back to the same thing. It starts with this smoothing here, right? Let's try to understand what happens here. And this smoothing is what is leading to your mating system, right? So again, yeast can access in two ploidy states. You have haploid cells, which we just saw. Okay, and then the haploid cells can have also a diploid state, which is forming because of the pheromone signaling. And these pheromones can come because of multiple reasons like deprivation or some other cellular key feature. And then you have these particular pheromones which come out. And then these pheromones are ligands. So they will bind to their receptors, and the receptors will initiate a pathway that enables them to fuse with each other. Okay, so you have, so now when you have this nutrient deprivation, which requires the cell to mate, the first thing that will happen is the cell will arrest themselves in G1. So they otherwise would have progressed to duplication and replication, but because there is deprivation of nutrient, they will first try to repair the cell. They will first try to address the problem at hand. So they will arrest the cell in G1. Then there will be a polarized growth towards the partner cell. Instead of bud formation, there will be as you see here, this thing. So the cells will try to move towards a partner cell. Okay. And then they will have smoothing. So they will have this projection called smooth that will be basically very nicely. These cells extend some projections that come into contact and pull each other close. Okay. So that's what will lead to this phase here. Here. Okay. So there you will have the cell walls fusing. Cell walls done plasma membrane fusing, plasma membrane done cytoplasm fusing, right? So ultimately, after all of these fuse, the nucleus will also fuse. Okay, so there, because your nucleus have also fused now, you will start having a nucleoid cells. And from these diploid cells, you will have a budding, and then you have two cells. One is daughter cells, which buds off, and then this one is remaining here. Okay, so now you have two cells coming out of here. So you have mitosis. So one is processes mitosis that we just discussed. This meiosis one, what happens is you have the sporulation happening. So this sporulation, sorry, just a second. This thing here, okay, the 2N cells will undergo meiosis if the stressful conditions are overridden. And then this will undergo sporulation. And then each of these cells will have the probability of going budding. So the main idea is they will be under haplotype spores and then they will give rise to budding. But if their environment is not proper, they will undergo sexual reproduction. Budding is asexual, this one is sexual. Okay, and this I told about two types of pheromones, MET A and MET alpha, that basically is responsible for their mating type. Okay, so which one they mate with. So you can have a MET A changing to MET alpha. It's basically which cells come together. For homologous recombination, that's like I hope that is you understand it with the questions more, because I think I should move to the questions and I'll explain it with the questions as we go ahead. Right. And as I said, this is a model organism. So anything that you study in the East is then used for understanding the process in other animals. For example, you can read a study about cell physiology changes, you can do drug testing or sequence variants or anything of that sort. Okay. Okay, so let's look at the questions so that we can end this session. So first of all, easy question, what does chromatin cerebral seed cells are, how small they are? Now, in budding yeast, each kinetochore binds to how many microtubules? Which complex links the kinetochores to the microtubules? Here, it's easy, but remember I told you last time also, the names of the proteins, the function remain the same. The names might change according to the system. So that's why the question here, what is the name in yeast? You know what we are referring to, but what is the name in the yeast? And which of the component provides structural strength to the inner layer of yeast cell? So what is basically present in the inner layer? Okay, so I'll give the answer. So the size of the cells is 5 micrometer, which we have discussed. So in eating yeast, budding yeast, you have each kinetochore, which is basically how this DNA moves apart, is bound by one microtubule. Uh, the kinetochore links to the microtubules. Kinetochore on the DNA links to the microtubules by NDCAT. We had kinetochore cohesin here otherwise. And which of the following components provides these, which present in the inner layer is the beta glucans and chitins. The other one had manoproteins. Okay, next set. During budding in Saccharomyces cerevisiae, which protein acts as the master regulator of the cell polarity? I told you where does the bud form? It's because of one protein. What structural features on the yeast cell wall indicates previous cell division? How I told you about the remnants of cell division. There are cues that the cell has undergone previous cell division. During mating type switching in yeast, the HO endonuclease creates a double-stranded break and which locus, we'll discuss that, and which genomic regions in budding yeast are typically associated with heterochromatin-like environments. Okay, coming to the answers, just a second. Right. Okay, so during budding in Saccharomyces cerevisiae, which protein acts as the master regulator? CDC42 that we have discussed already. Okay, the next question. During mating type switching in yeast, the HO endonuclease creates a double-stranded break in which look at MAT, because remember MAT alpha and MAT A are the ones that govern the fusion, and it's the opposite. So MAT A will bind to the alpha, the MAT alpha will bind to the A receptor. So that's the switching that we are talking about, right? And then you have the bud scars, which are present on the cell wall to say that indicate previous cell divisions. And the in the budding yeast, if you have regions which are closely or which are analogs of heterochromatin like environment, these are telomeres, rDNA, and silent mating loci. These are all very characteristic feature of yeast, right? So heterochromatin and euchromatin difference we know. Similarly, whatever functionality we associate with heterochromatin, these are the regions in yeast that we associate the functionality with. Okay, and the last one is in the East-2 hybrid system, expression of Laxzi reporter occurs when? What is the primary purpose of East? What is the importance of East-2 hybrid system? And during the apical isotropic growth switch, what major change occurs in bud? And what is the approximate size of centromere in the East? Okay, one more minute and I discuss the answers after that. Okay. So, so in the E2 hybrid system, expression of laxi reporter occurs when two proteins of interest interact and bring activation and DNA binding domains together. Right? I will explain to you in a bit. Primary purpose of the system or the method or the technique is to identify protein-protein interaction. Unique apical and isotropic growth switch. Okay, you have apical, which is bud formation, and then you have just equal division across the cell surface. Uh, what major changes occur in the bud? Growth becomes uniformly distributed over the bud surface. So first you have just the apical growth, which is basically in the entire cell, you have just one apex, one side, which is growing to form a bud. And after that bud is formed, then you have these two surfaces, the growth becomes uniform across the bud surface, right? So this is the original cell. You have this small bud forming here to make that bud prominently outside the cell surface. And then you have isotopic growth, which is basically everything going together. And the centromere is around 125 base pair. So quickly, I will just explain the East2 hybrid system, which came up in these questions. These are basically protein-protein interaction, as I mentioned. And there are two terms that we mentioned here, right? DNA binding domain and activation domain. So basically, the whole technique is basically relying on the idea that the transcription factor generally have two separate functional domains. So you have a DNA binding domain, which binds to the DNA and says, okay, this is the DNA sequence where your expression has to begin, your transcription has to start place. And then you have an activation domain, which activates the transcription of the reporter gene. So it doesn't just bind, it then activates the reporter gene. So brings in all the factors that are responsible. So the idea here is that since these domains cannot activate transcription independently, they have to be always together for coming together and doing the function. So in your protein setup also, we always put this bait protein and a prey protein. One is with the DNA binding domain, one is with the activation domain. And what we want to do is we basically put the bait with the binding domain, uh, make it fuse uh, to the promoter region of the LAC-Z. Okay. And then, so it has binding domains, it is bound to the LAC-Z. Now, if the prey protein, which is what we are detecting if it binds to the bait protein or not. If the prey protein interacts with the bait protein, then the activation domain is brought close to the promoter and the transcription factor becomes functional and the reporter gene is transcribed. So I hope you understand this. You have LAC-Z. For activating LAG-Z, you have a DNA binding domain and an activation domain. You put both of these in two proteins that you suspect are interacting. In one, you put binding domain. In one, you put the activation domain. If these proteins interact, they will come close to each other and activate the LAG-Z. Why LAG-Z? Because LAG-Z gives us a visual output. LAG-Z basically produces beta galactosidase. And when you have beta galactosidase, your colonies become blue in the presence of your inducer, which is XCAL. So if you see blue colonies, you know your proteins have interacted. If the proteins don't interact, you need both the domains. If the proteins don't interact, the both domains don't come together and the gene doesn't get activated. So that's the system that we use to understand the proteins that we are trying to understand based on their interaction. We come to the conclusion based on this analysis, and this analysis is happening because in yeast we know that the transcription factor have two domains. So it's just one of the applicability of the yeast for molecular studies, molecular understanding, right? Okay, so that brings us an end to this session. Next session is Monday, usual timing, and that would be the last session for this course. And we would wrap up the course, go over the topics of that week, and I will just not describe, but quickly just give you a mind map of what we discussed over the past 8 weeks. And hopefully that will help you in your upcoming exams and clarity of your doubts, right? Okay, so we stop here today and we meet each other in the next session, which is on Monday, right? Okay, so any doubt you can mention or else we meet on Monday.